Yeah.
Incredible.
Every day in the world, agriculture feeds 7 billion people. And this number will reach at least 10 billion within the next 50 years. Agriculture is the source of our food. Everything we eat depends on it. Agriculture produces vegetables, fruits, and cereals, but it also feeds the animals that we eat. The bad news is, agriculture is in real danger. A United Nations report claims that an increase of 1 degree in temperature could decrease agricultural yield by at least 2%. We'll need more fields, but centuries of urbanization have left no space, unless we destroy precious nature areas like rainforests. Even worse, since the 1900s, more than 90% of the fruit and vegetable varieties have been lost. Why is the loss of diversity such a bad thing? Did you know that today more than 60% of the world's calories come from 3 crops: wheat, rice, and maize? While those crops are originated from specific places in the world, they are grown everywhere, from East Asia to the Americas. As they spread, they had to face new challenges, but they also diversified. Quickly, farmers and professional breeders realized that this biodiversity could be used to create new varieties more resistant to cold or drought and to increase yielding. This is why preserving crop diversity remains the best way to help agriculture adapt to the challenges and demands we face. Luckily, crop diversity, though in danger, is not entirely gone. Farmers still maintain it in their fields, while scientists have established hundreds of gene banks in the world to safeguard this diversity. We at the Crop Trust are here to help these gene banks. Our mission is to create a global system with secure funding, efficient management, and coordination between the world's gene banks. Thanks to this global system, over the last 10 years, we've already secured more than 700,000 crop varieties in international gene banks. And we also made sure that these varieties were backed up in the Svalbard Global Seed Vault. But the work is not done yet. We need to secure our agriculture diversity forever. We need to build a self-sufficient global system encompassing the whole of crop diversity. This is why the Crop Trust is raising an endowment of $850 million that will produce the funding required on an annual basis, indefinitely. But we can't do that without your support. Help us make sure our children and theirs will have enough food to eat. Together, we can secure the foundation of our future. Together, we can secure our agriculture, our food, forever.
Welcome, everyone. My name is Faye Wambuel-Ludeling, and on behalf of the Crop Trust and the GeneBank platform, thank you so much for joining us for today's Grow webinar. Just a little housekeeping before we get started. Right now, we have everybody on mute so that we can avoid background noises that may distract you from listening to this webinar. However, we'd love to hear from you. If you have any questions or comments during the presentation, please type them into the Q&A box in your Zoom control panel. We have a team dedicated to managing this functionality throughout the presentation. A recorded version of this webinar, as well as a presentation deck, will be made available immediately after this presentation on the GeneBank platform website. I will now hand over the session to my colleague, Yanni Van Beem, our moderator. Over to you, Yanni.
Thank you, Faye. Welcome all to the second presentation in the Grow webinar series. It's my pleasure today to introduce Professor Sophien Kamoun. Sophien grew up in Tunisia, where he developed a passion and curiosity about nature. He studied genetics in Paris and Davis, California, before working in Wageningen, Ohio, and Norwich, where he is currently a senior scientist at the Sainsbury Laboratory and professor of biology at the University of East Anglia. Sophien is known for his seminal contributions to our understanding of plant diseases and plant immunity, having pioneered genomics and molecular biology methods to reveal fundamental insights into the evolution of eukaryotic plant pathogens. His inventive work in plant pathology has resulted in new approaches to mitigate some of the world's most serious crop diseases. Professor Kamoun has received many awards and recognitions, notably the Kuwait Prize and the Linnean Medal. He was elected a Fellow of the Royal Society in 2018. The title of Sophien's talk today is CRISPR Crops, Plant Genome Editing Made Easy. Welcome, Sophien.
Thank you. Thank you so much, Yanni, for the introduction. Let me slides here. And thank you for the invitation. Thank you, Faye, for setting it up. I was tasked by Yanni to talk to you about CRISPR and gene editing, but in the context of conservation of plant genetic resources. And I really have to thank you, Yanni, for the invitation, because I didn't give this topic as much thought before as probably deserved. And so this really prompted me to do a lot of reading and catch up on this. So I will not be talking to you as an expert, but really as someone who will kick off a discussion. And I hope we can have a really good discussion after my presentation and in the future. So here's the plan for today. So the plan is I will first introduce CRISPR. I give you a bit of an overview of the technology and what it all means. We'll go through some of the applications of CRISPR gene editing to plant biology, plant breeding, and then we'll finish with the topic of how does CRISPR and conservation biology intersect. So obviously we've all heard about CRISPR, hopefully more in good ways than bad ways, but you know, there was also some bad press. I mean, the whole disaster of the CRISPR babies wasn't the best use of this technology, but there is no question that this technology is game-changing as far as plant breeding goes. And this is why, because we've been improving crops, as you know, ever since humans domesticated plants. And if we think of the genome as a book, then some of the early types of improvements were based on hybridization. So that would be like bringing volume 1 and volume 2 together, two different books, and kind of merging them into one. Introgression is more like moving a chapter from one book into another. Mutagenesis would be randomly changing text in the book. And transgenics, of course, is about moving maybe sentences or a paragraph from a very different book into a new one. So where does CRISPR sort of fit in this continuum of genetic improvement of plants? Well, the nice thing about CRISPR, if you use that metaphor of the book, then it's the ultimate in terms of precision, because now what we can do with gene editing is we can change one letter, we can change a few letters, we can remove a few letters, and we can do it precisely in exactly this specific location in the book that we wish to target. So essentially, it's a little bit like a word processor. And one of the sort of, I think, exciting thing for me as a geneticist is that this is it. This is the ultimate in precision. If you can go to a book and modify the specific letter you want to modify, then we cannot do better than that. So obviously, the technology is very early, and some applications are easier than others, but ultimately, this is it. If we can do that, then we can now sort of think beyond, in fact, beyond genetic improvement, and start really thinking more about the application than about how to achieve these changes. The second point I want to mention, and I think it's sort of the history that led to CRISPR. So CRISPR is not the first time we could do reverse genetics. So reverse genetics, for those of you who need a refresher, is essentially starting with the gene and going back and making a mutant plant, right? So this can be done with mutagenesis, with tilling, with insertion mutagenesis, whether through agrobacterium, tDNA, or transposons. There are various ways of doing gene knockout, gene knockdown, sorry, using some various forms of RNAi. And also there were methods for gene editing before CRISPR. So zinc finger and talons are two very good examples that are also enabled gene editing. What was really beautiful about CRISPR, what was really game-changing, is the fact that it's so easy to implement. And this is really why it took biology by storm, because all of a sudden, pretty much anybody with a reasonable molecular biology lab could do gene editing in whatever organism they study. So I think it's worth going through a little bit the technology here and in a little detail, and really, especially for the students or early career scientists in the audience, if you haven't done that, please read as much as you can about sequence-specific nucleases, because this is really the key to this technology and really a key player in terms of future genetic manipulation of organisms. So essentially what this means is that you have a nuclease that will cut somewhere in the genome at a very specific location. And the beauty of these nucleases, like the zinc finger and the TAL effector nucleases, and also the CRISPR Cas9 enzyme, for example, is that they will cut at a specific location that you could actually guide the protein to do so. So they are programmable. They can be programmed to cut any sequence in the genome that you wish to target. And the reason why CRISPR really was game-changing compared to the other SSNs is that the programmability, if you like, the specificity is done by an RNA, the so-called sgRNA or small guide RNA. And that RNA is much easier to deliver to the plant than changing the entire protein, which is what is necessary for the zinc fingers and the tannins. So this guide RNA, that defines the specificity of CRISPR, is really what made the technology so easy, much easier to implement. And that's really the big advance here with CRISPR, is just much easier. That's why I like to title my talk, Gene Editing Made Easy. So the CRISPR craze really took off in August 2012 with this publication, which is, as you can see here, really describing that there is a guide RNA that is programmable and can be modified to target any sequence you wish. And this is the paper that led to the Nobel Prize to these two wonderful ladies, scientists, Emmanuelle Charpentier and Jennifer Doudna, who just received the Nobel Prize a few months ago for a discovery that was only made in 2012. So as far as really biology goes, this is truly a craze, because this is quite unusual to have such recognition and such adoption of a technology just 8 years after it was invented. It was really crazy because very soon after that paper, I mean, this is what, few months after January 2013, there was already applications in humans for basically using the system, the Cas9 CRISPR system to make targeted mutations in human genomes. And then the CRISPR craze also infected plant scientists. That was not long after, really essentially a year after that Nobel Prize winning paper, where many of us in the community implemented and showed that CRISPR can be used quite easily actually in plants. Okay, so what can be done with CRISPR? I don't want to take you really through all the details here. This is more really as a reference for those of you who want to find out more. I really encourage you to read this particular review article by Kaxia Gao, who is one of the pioneers in CRISPR technology. She's based in the Chinese Academy of Science in Beijing. And the three methods really that are worth thinking about here are gene knockout, where you basically make a loss of function mutant. You basically take the gene out of action. Gene knock-in or replacement, which is when you bring in a new sequence or a modified sequence of a given gene in a specific location in the genome. And then the third one, which is perhaps the most interesting in a way, is really base editing, where you're truly doing editing. You're changing now the genome text in a way to a different type of text. And this can result in changes in amino acids, small deletions that are in frame, not necessarily totally fully disrupting the protein, but modifying it, and so on and so forth. So just really a few examples for those of you who are more technically inclined is you could also have two targets in the sequence. So you can deliver one guide RNA or multiple guide RNAs. And if you deliver guide RNAs that are adjacent in the genome, you will make two cuts in the genome. And that's essentially a fairly straightforward way of making a deletion. In fact, this is probably the simplest application of CRISPR, is to make a deletion and essentially remove a piece of the genome from that particular plant. So in this case, as you can see, you have two targets here in the bottom part, target 1, target 2, they're shown in red. And then by using the CRISPR system and delivering these two guide RNAs, targeting these two sequences, we introduce these deletions, which are about 70 or so in base pair in length. And so CRISPR can be used to make small deletions, like you can see here, tens of bases, tens of letters in the genome, but can also be used to make big deletion. In fact, this is, I think, our record so far is a deletion of 53 kb in a tomato cluster with, as you can see there, with 4 different genes in that cluster. And that whole cluster of 50 kb, 53,000 bases, 53,000 letters was actually totally deleted in this particular tomato mutant. So these are sort of some of the applications of CRISPR, and I again don't want to really spend the whole time discussing that. There's quite a lot of variations on this method right now. The interesting point really here is that we can accelerate the process of mutation breeding. So mutation breeding in the classical way is based on using chemicals or radiation to introduce random mutations in the genome. And then the challenge became finding essentially the needle in the haystack, right? So you want to find, you have now this big population of mutants, you're interested in a particular gene, and you need to find that mutation. And I have to say also that this has a big role to play in plant breeding, and it is actually greatly enhanced and facilitated by genome sequencing and so on. So there are many tricks now to find, for example, that needle in it was just a few years ago, thanks to genome sequencing, for example. I really want to emphasize this point, because I'm not here to tell you that CRISPR is sort of a magic wand that's going to change everything. It's a fantastic technology, but there are also other technologies, especially genomics assisted or genomics-based selection, which are also really having a huge impact on plant breeding. And I think CRISPR is part of that continuum of methods. It does indeed accelerate things, but it's again another tool in the toolbox, an exciting tool, but nonetheless, it is complementary to some of the other methods. Okay, so I'm going to open a parenthesis here, and before I continue with the talk, because I'm sure this question will come up. It comes up all the time, and it's on everyone's mind. What about gene editing? Is it GMO? Is it not GMO, etc.? So oddly enough, my colleague Eric Ward and I, back in 2012, actually before CRISPR, we wrote this commentary that at that time it was really based on some of the first application of TALEN type gene editing. And the irony here really is that when you make deletions, like I showed you that example of that tomato with essentially 53 kb of the genome removed, so 4 genes are actually removed from that plant. In a way, this is the exact opposite of what we do with GMOs in the sense of transgenics, right? Because transgenics really are about bringing new genes into your crop. In this case, we're doing the exact opposite. So I did want to bring the issue of whether CRISPR is GMO or not, but I also want to emphasize that this is not really a scientific question. CRISPR is just part of that continuum of genetic modification we do through breeding, through mutagenesis. You can actually use CRISPR to introduce naturally occurring mutations in a different crop background. So in terms of the product, in many cases, in most cases, gene editing is not actually producing something that is dramatically different from what you would obtain through natural methods or classical methods of plant breeding. So in a way, the question of whether gene editing is GMO or not is more of a regulatory question rather than scientific one. And that varies depending on the countries, because different countries have different approaches to regulation. Some are process-based and others are product-based. So without spending too much time on this, I just want to highlight that there is a lot of diversity currently in how different countries regulate gene editing. But it's worth noting that some countries that are traditionally perhaps more conservative, like Japan, for example, have decided not to regulate targeted mutagenesis or gene editing in the same way that they regulate GM or transgenic technology. All right. A second point I want to make in terms of my parenthesis is point out to an example of a country that has fully embraced biotechnology in general and CRISPR in particular, and this is China. So this is a company, Biogle. That young fellow on the left there is actually the young founder of Biogle. This is a company based in Changzhou. I had the opportunity to visit it a couple of times. Changzhou is somewhere between Shanghai and Nanjing. And this is a company specialized in CRISPR technology, in particular in plants. And this is their lab, and actually all these scientists there are, when I was visiting, were at that time doing gene editing in maize. So Biogle offers this as a service to the Chinese scientific community, and I suppose to industry too. And they do CRISPR, again, not just in plants, but also in other organisms. And they already have published quite a lot, and they're sort of the outputs of the libraries that Biogle has produced already in dozens of papers. So this is an example that dates back already to 4 years ago. This is 2017. Their first, I think, large-scale library of plant mutants. This is actually rice library. I forgot the number of genes here, but it was maybe half the genes in the rice genome that were knocked out and present in one of those blue boxes. So any scientist who would like to obtain a mutant in a specific gene just needs to order it through Biogle, and then they sort through their gene bank and send them the mutant. And by now, they actually just recently published a similar library for maize. They've done soybean, tomato, obviously model plants like Arabidopsis and Nicotiana. But I mean, this is something that is already happening in a massive scale, and possibly already introduced into the breeding pipeline through the material generated by this one company. Okay, so back to the applications. Again, I don't really want to do a thorough job, but I want to highlight perhaps the applications that I think are most relevant to your group and to this to the topic we are concerned with. So again, I point you to this review article if you'd like to know more. So one of I find actually the most fascinating perhaps aspect of CRISPR is the capacity of generating allelic series. And so this is shown here, for example. So this is work from my colleague next door, Jonathan Jones. He took the gene DELA, which is actually a famous gene in plant breeding and plant science, because this is the gene that's underpinning the green revolution. This is the DELA gene. Mutations in the DELA gene produce dwarf plants, whether it's wheat or tomato. And what Jonathan's team did is they targeted DELA and made a single cut here in this position. And so what that does is when you use the CRISPR system to make that cut in the genome, what happens is mutations are introduced randomly at that position. So you could have small deletions, you can have changes in the sequence, you can have insertions, And this is just an example of some of the alleles they produce. So they had one, the first one here is a T insertion. So there was an insertion of the letter or the base T. The second mutation is a deletion of 5 bases. And the third one here is a deletion of 3 bases. Now, what's really interesting is they targeted actually the specific motif that is known to be important for the function of this protein. And if you have, within the coding sequence, you have a deletion of 3 bases, then what that means is you end up having a protein that will be coded and will be very similar to the wild type protein, except for the absence of one single amino acid, because 3 bases corresponds to a single codon. And that very specific allele is quite unusual, quite specific, and indeed turned out to be a very interesting allele of DELA, because that particular mutant that was missing one amino acid turned out to be coding for this dwarf or semi-dwarf phenotype that is actually a dominant phenotype. So you could see here that with using CRISPR, you could actually generate an allelic series in the gene you're interested in, and screen also phenotypically for mutants that might be interesting or might be favorable. So it's a very interesting, I think, concept, because if you think about it now, what Jonathan's team did here is they generated an allelic series, and they had many more mutants than these. This is just an example of some of the mutants they're showing. But they generated a very long allelic series in DELA that is perhaps beyond anything that was generated before. And so in a way, what gene editing is doing now, it's accelerating our capacity to produce biodiversity, to produce new alleles, new plants, perhaps with really interesting phenotype at a scale that is beyond what we could do before that. Or at least you could do it, but then you'll have to really generate millions of plants, millions of mutants to really be able to get the same type of allelic series. So that's one really interesting application. Now, my field is plant pathology, so I have to tell you something about applications of CRISPR to plant health. And one simple concept in plant pathology is that pathogens require host targets to enable them to colonize these hosts and cause infection. So in a way, the pathogen has a key, the host has a lock, and if the pathogen can unlock that lock, then they can cause infection. So one strategy in CRISPR is to modify the lock, or even sometimes remove the lock. In that case, the key doesn't work anymore. So this is what we call susceptibility genes, or S genes. So basically, host genes that are enabling the pathogen to cause disease, and essentially the lock and key concept. So one way to do produce actually resistance is to mutate these S genes or susceptibility genes. Some cases make a full knockout, essentially make a non-functional version of that gene. So this is our proof of concept. We use tomato, and we use resistance to the powdery mildew fungus. And there is a gene known as MLO, which is known to confer resistance or to confer susceptibility, should I say, to the powdery mildew fungus. So we use CRISPR to make that double cut and make that precise 49 or so deletion in the genome, in the MLO genome, and indeed the plants were fully resistant. So this is perhaps the simplest application of CRISPR for plant health, is to mutate or knock out susceptibility genes to make the plants more resistant or less susceptible to pathogens. And there are, again, many applications, and the creativity of the scientists is really enriched with this technology. This is an example also from my colleague Zach Lipman at the Cold Spring Harbor. What Zach did is he essentially produced through the introduction of 3 different mutations in tomato, a sort of a dwarf bushy tomato that he's promoting as being ideal for urban gardening and balconies and this sort of thing. So it's it is quite really interesting. I think that people are thinking of this type of applications, and this is quite straightforward. This is really one single gene cassette with 3 pairs of guide RNAs, and then that's enough to produce this mutation. So Julian Shao in Maryland, he did this last summer with his students, and that was a summer project, I think, for the students as part of the biotechnology program. And guess what? When they planted these dwarf tomato, they got powdery mildew infections. So now he promised me that next year they will actually include 4th mutations, which will be that MLO mutation I just showed you, to make not just this dwarf bushy tomato, but also make it powdery mildew resistant. So you can see again here the powerful combinatorial effect of the technology, or the capacity of the technology, where you could actually combine all these different mutations, just keep adding essentially. And so it's really up to the creativity of the scientist to produce the plant they want. Another really incredibly creative, I think, application of CRISPR is de Novo Domestication. So this is something that started just a couple of years ago with tomato was the first example, where by introducing 5 different mutants, you end up having essentially taking a wild tomato species and making it closer to a domesticated, as close as you can to domesticated tomato. And of course, this is really interesting in terms of breeding, in terms of bringing in new traits and all that. And more recently, this is just 2 weeks ago, the same idea of introducing domestication alleles, essentially, if you like, into a wild species of rice, so turn it into more of a something more similar to cultivated rice. So this is, I mean, this is again just the beginning. So you can imagine here the speed at which one can be creative and generate maybe interesting applications for the future. Okay, so let's see how we're doing with time. Okay, good. So now I think we can get to our topic of interest and the role of CRISPR and gene editing in conservation biology. And I'd like to start by pointing out to this really interesting review, which was beyond plant genetic resources and crop conservation, but really talking more generally about conservation biology, and pointing out that really the field of conservation biology does require a bigger toolbox to meet these challenges we have in this century. So climate change, habitat loss, invasive pathogens and pests. We know that has increased quite a lot in recent years. These are really big challenges, unprecedented, and they require perhaps a bigger thinking and sort of considering incorporating tools of biotechnology into this field of conservation biology. The next point really is that when we talk about conserving germplasm, we should also think about knowing the germplasm better. And I think conservation genomics is absolutely essential and goes hand in hand with conservation of plant genetic resources. If you think about, we're at such an early stage in gene editing that really in the future.
I didn't get that. Could you try again?
Sorry, I said something that triggered Siri on my phone. But, so I think the point here really is that if we sort of fast forward to 50 years from now, and maybe editing is really widely used, what would be perhaps more important then is having the sequence and knowing what the valuable alleles and mutants are. Perhaps that's even more important or as important as having the seeds themselves. Given that then the technologies, the breeders can introduce those mutations into their material, the plant material. So I think the importance of genomics and scaling up our sequencing of crops is really important. And then, of course, there's this idea of using gene editing to facilitate adaptation. We know that the climate, for example, is changing. So many of the land races we have are not perhaps becoming as well suited to the host climate as they used to be. So maybe we should also consider using gene editing to improve land races and really make them handle the changing climate. And then there's also this field of de-extinction, which is a really fascinating application of gene editing, and I'll tell you in a bit about that. But this is where we can even go to extinct material, extinct crops, where we have perhaps herbarium material or tissue that is stored, and identify against the alleles, the variants, and then introduce them back into modern materials. So essentially resurrect genes and sequences and alleles and variants that may have gone extinct by introducing them into more modern or extant varieties. So these are sort of the thoughts I came up with when I was researching this topic. And just maybe a few comments before we engage in a discussion is the first one really is we should appreciate that crop genomics is really, really new in a way. For example, if you take something like wheat, which is perhaps the most challenging genome to sequence, really the first somewhat complete sequence of a wheat genome was only published, what, 2-3 years ago, 2018. So that's the first really genome sequence where you have the different haplotypes separated, different genomes separated, and then a somewhat complete sequence. So that's really recent. And then just 2 years later, you have the first, not really pan-genome, as we say, but already a very good documentation of the variation, genetic variation in the wheat genomes in the modern cultivars. So again, these are really incredibly useful resources for the futures in terms of identifying the valuable traits and alleles that could be incorporated into modern varieties. So I again insist on the importance of conservation genomics and how maybe your group can work more closely with initiatives that are out there in terms of sequencing crop genomes. And then there's also this idea of essentially going for what's extinct or what's endangered, and this group that George Church, who's a really visionary scientist and biologist at Harvard, who has this initiative called Revive and Restore, where the idea really is going for endangered or even extinct organisms and having here a framework for how to really address or enhance their capacity to survive or even revive them. So talking about facilitated adaptation is a really interesting concept. So if land race that is maybe facing a drier environment, less rainfall, maybe could be improved by making that land race more drought tolerant, for example, something like that. There's even de-extinction, which is something really fascinating. So if you go to their website, you see that they have even a project on extincting, I guess, or whatever that verb would be, the mammoth and Przlowski's horse and even coral and so on. So this is truly a visionary view, but although it may sound like science fiction, this is something we're really inching closer towards. So those were really my thoughts. And I again want to insist on the fact that this technology and this tool is really new. It's in infancy, despite, you hear about it all the time and there's a Nobel Prize and all that, but really CRISPR was only invented in 2012, 2013, as far as plants go. So we're talking about a very young technology and we haven't yet explored all its full potentials. So I think it's important to end with this quote here, which I really like, and I find it very appropriate for this topic. I didn't come here to tell you how this is going to end. I came here to tell you how it's going to begin. So I think we're really at the beginning of the applications of gene editing to to agriculture and crop biology. So with that, I'm happy to start the discussion, but I really want to thank some folks in my group who have been long intellectual and wonderful intellectual friends and wonderful colleagues. In particular, Vlad Nekrasov, who now has his own group doing gene editing in wheat at Rothamsted Research, and various others, more recently, Torsten Langner, who were leading some of the CRISPR work in my group. So with that, I think we have time for a good discussion. And I see that there are quite a few questions already, which I couldn't read yet, but I'm looking forward to your questions and your feedback. Thank you very much.
Thank you, Sophien. Thank you for that very comprehensive walk. Simplistic for those of us that don't work with the system, but I think it opens up to new possibilities and takes us up from producing more diversity into what can be done in future with maybe extinct creatures that are no longer here. We do have a number of questions. I'll start from the very top. First question from Nora Castañeda. Are there examples of commercial varieties improved with CRISPR?
Good question. I don't know if anything has fully made it to the market in terms of crops. I know there are certainly mushrooms and quite a number of things in production, but there are certainly other examples of gene-edited crops that have made it to market. So canola, for example, there's herbicide-resistant canola, which was using a different technology, pre-CRISPR. But my understanding is that this is really accelerating now because of the regulatory, the sort of more flexible regulatory framework that is being implemented in several countries, notably in the US, actually.
Okay, thank you. The next one is from Benjamin Killian, who is here at the Crop Trust. As part of the Crop Wild Relatives project, we are coordinating pre-breeding activities in 19 food and forage crops important for food security, including finger millet and grass pea. What is required to perform genome editing in such neglected crops? And then he lists some things. He says availability of reference genome sequences and pangenome or transformation, possible or effects known for target alleles and haplotypes. What else?
Very good question. And thank you, because this really complements one of the messages, one of the take-home messages, which is, yes, first step is having the genomes. So really, again, I really want the community to think about sort of crop conservation genomics, if you like. If I were sort of to really be very concerned about a particular resource or particular crop that's neglected, valuable land races and all that, I would really start sequencing it. And that's becoming more and more accessible with reduced cost and all that. So that's step one, you need the genome. The second thing is transformation, and transformation varies because in many cases you have even if the crop can be transformed, sometimes this is not actually the variety you're targeting or the landrace or the local variety that is transformable. So advances in transformation technologies are also important. There are some very exciting advances. I didn't have time to cover that, but we can discuss that. There's some recent work that is really opening the door to improve transformation methods. There are also different ways of transforming crops, not just through agrobacterium, but there are other methods. So that's also something really exciting. And then the third part, I would actually reframe your third point, which is totally irrelevant. Obviously, you want to know which mutation to introduce. I would frame it into more sort of a basic knowledge. So I think this also brings us back to more investments into fundamental research in terms of understanding the traits and understanding the genes that are valuable to agriculture. But in many cases, even if it's a neglected crop, let's say finger millet, for example, then there's a lot of knowledge from rice or wheat that probably can be translated to finger millet. So there would be enough. If you want to make a drought tolerant or salt tolerant finger millet, there's probably enough knowledge from other grasses that are better studied to bring to target similar genes and introduce similar mutation. In the same way, actually, we did it with that tomato MLO. MLO was discovered in barley, so we basically translated the knowledge from barley to tomatoes. So a lot of times, neglected crops can benefit from that. But that's fantastic. Your summary there really does cover the main issues and the main things you need to bring this technology to neglected crops. Thank you.
Okay, thank you. The next one is from Babar Usman. Sometimes single gene mutants do not show expected phenotype. This may be due to the gene redundancy. This is very difficult sometimes to find the genes which are redundant to each other. How to overcome this problem?
It's a fantastic question, because that's actually one of the exciting things about CRISPR is it opened the door to study redundant genes. In fact, that's a topic I study because we work on an immune receptor and our gene network, and there is redundancy in that network. And without CRISPR, it was really difficult to dissect that network. So now we have actually that example I showed you with the 53 kb deletion, where we knocked out 4 genes. In fact, 3 of them are redundant. So that just answers your question. So that's something that was very, very difficult with, let's say, with tilling or with chemical or mutagenesis or radiation. Is really extremely unlikely that you'll get double mutant. But with CRISPR, by targeting it, you can actually target an entire gene family. So this is an application I didn't get into, but indeed, this is something absolutely exciting and opening our minds really as geneticists to the complexity of genetic systems and gene networks.
Wonderful. We have another one from Tom Payne. What is the need to conserve native genetic diversity for the future with the advent of these new biotechnology tools? I think that is the key question in all of the gene bank managers is what is the need to conserve? If you're saying you can produce a lot more diversity now in the laboratory.
I think these things are complementary. I really hope that this technology will further enhance the value of the genetic resources and the gene bank and all that. Again, I think a lot of these plants are, I think the most valuable thing to me, of course, is that these plants are adapted to their environments. They've gone through thousands of years of domestication. So they're great templates. I mean, the reason, for example, let's take the Novo Domestication exercises and projects that are really sort of mind-boggling. I mean, really, you could take a wild tomato and sort of make it closer at least to something that you could cultivate. This is mind-boggling, but let's not forget that that's actually translating the knowledge we know about the domesticated tomato or the domesticated rice and applying it to, in sort of a fast-forward type way. So the value, I think, of what we created as humans in terms of germplasm, in terms of domesticated crops, in terms of also all the land races and all the locally adapted varieties is extremely valuable. There's no question that we need to conserve that, and we need to actually make sure that that doesn't go extinct. And I think what this technology do more than anything, they add value, like genome sequencing, for example, just can add extra value to those resources. You know, I really wish, I mean, I'm from Tunisia, so obviously 50% of the calories come from wheat. You know, my dream would be that, you know, while I'm still alive, that all the local varieties would be sequenced, all the local wheat varieties in North Africa would actually be sequenced, and they would be stored in a database. I think that would be, to me, that's as valuable as storing the seeds in a way, or complementary to storing the seeds. So I really wish that to see more of a merger again between conservation genomics and conservation of biomaterial.
I think we're starting with that with this talk, is the conversation between what you do and what we do, and how can we approach that. We have a question from Nelissa Hamora. What's your prognosis on the role of this technology to closing yield gaps and pushing yield frontier for food crops?
Oh my gosh, then Elisa, that's a really broad question, and I think it's so complicated and so context dependent. Oh gosh, I mean, it's I think there's obviously I don't want, it's not my field, so I don't really want to get too much into that. But you know, the big dilemma we have now, I know that from wheat in Tunisia, for example, is the introduction of these modern varieties is a complicated topic. It, you know, this displaces the local land races. You know, the farmers don't have anything to feed their sheep anymore. They need more, spend more money on fertilizer. It's a complicated topic. So if we could somehow go back to improving local varieties, if you had technologies that allow us to improve and accelerate the breeding of the local varieties of the land races, you know, and improve them in some ways that we couldn't do it before, I think that would be the best way to answer your question. I think the introduction of modern varieties and modern practices is good, but it's also complicated, as we know. It's a complicated topic. So I wish these technologies can sort of makes us take a harder look at the local varieties and the land races and see if there's potential maybe for improving them rather than bringing completely new foreign germplasm to a region or to a country.
Yeah, and actually related to that last one that Melissa posed, we have one from Lucas Buchholz. I'm hoping I pronounced that last name correctly. Isn't there a danger that beyond a certain degree of genetically modified plants, the natural balance in the biosystem would be destroyed? Or taking it even further, could vegetation in certain areas become dependent on scientists?
Uh, okay, it's a very broad question, and I think this goes way beyond gene editing. It's really applicable to any form of plant breeding or genetic improvement. I think the second part of your question, vegetation become dependent on scientists, I think it's very evident to me that crops are dependent on the farmers and on the plant breeders. So I think that's something that, you know, if many of these crops wouldn't survive in the wild without human intervention. So I think with domestication, we already started that sort of symbiotic relationship with some of these plants. The question about the natural balance is also, to me, I mean, again, I'm not a plant breeder, so it's hard for me to answer it directly, but I don't see farms as part of the natural balance. I think a lot of times farms are really human modified systems, and for example, I think if we want to look at compare our crops to the natural world, I think they're more like the pets we have, like the cats and so on we keep in our houses, than the wild tigers and the wild lions. So they are tamed, they are modified to be to be domesticated. So I think it's a very good question. It's a philosophical question that is really worth a very deep discussion. But beyond what I just said, I don't know what else I have to offer, really. I think it's an interesting question that goes beyond gene editing. What is the role of, what is the relationship between agriculture and the natural world, and where does that fit? Where do plant breeders and scientists fit in that balance? It's a good question.
Yeah, it's good to know your thoughts also and your comparisons that you make to what we've domesticated and what
So
we
I have a slide I kept at the end because I often get the question. I'll show it because it's a fun slide to show, and it's related to this topic because I often get the question of, let's see, I'm not sharing my screen now, right? I often get the question, well, how can you remove genes? You know, you're removing genes from the plant, but these genes are there for something. And aren't you sort of, how aren't you messing up with this plant by removing it? And what I like to show for that is this slide of a very ugly creature. Can you see my slide now?
Yes, your cat. Is this your
Exactly.
cat, Sophien?
It's not my cat. So yeah, well, at least in pre-COVID, I traveled too much. We traveled too much to really afford having pets here. But this ugly little thing is exactly what we are doing with our crops. I mean, our crops are already like that. They are, of course, this would not survive in the wild, but removing the hair for a domesticated cat, as they would do just perfectly fine in your flat or in your apartment or your house. So it's a bit like that. So of course, these genes are really important when these organisms are living as wild plants in the natural environment. But that's not what our crops are. They are pampered, just like our pets. And so we should not forget that, I think.
Yeah, that's a good point. Moving on, Matthew Ruard, more in your field now. Are you aware of publications reporting modifications on a specific gene on all homologous chromosomes in a polyploid plant? Does CRISPR-Cas9 edit allelic variations at once if high similarity?
Yeah, so the question, Mathieu, is very simple. It's yes. It has been done in wheat, for example, where all 6 copies were mutated, and in some cases there are families. So actually, CRISPR is really efficient. So if you have multiple copies, very often you'll get the multiple knockout. So if you send me an email or something, I'm happy to point you to publications. But it has been done from the early days. So this is relates to that question of gene redundancy in a way. So you could actually mutate multiple alleles or homologs, or you could also mutate paralogs. So it's a fantastic technology indeed for targeting related genes, multiple related genes in the genome.
Yeah. Next one is from Jeffrey Houghton. It's a 2-part question. The first is, I don't understand the argument for why deletion is not considered genetic modification. In other words, does not result in a genetically modified organism. Please, can you explain further? And I'll give you the second part afterwards.
Yeah, sure. No, that's a very straightforward answer. I mean, it depends how you define GMO. I think we're using the definition of GMO equal transgenics in the sort of the common understanding of what the GMO is. Scientifically, GMO, genetic modification, doesn't mean anything, right? I mean, we're modifying genes and in using various methods, including classical breeding. So genetic modification here, as in transgenics, where you bring a gene from one organism into another, in that case, that's the exact opposite of removing a gene from the host genome. That's what I meant by that.
Yeah, the second part, it's been placed somewhere else. The second part, yeah, there it is. Some of the countries that could benefit most from gene editing do not allow its use. How do you see this situation being addressed?
Oh, I'm not sure that. I mean, I think right now the biggest so the biggest area where gene editing is not being used is Europe. I think otherwise, outside Europe, you either have countries that are sort of loosely regulating gene editing, certainly not as strictly as transgenics, and you have countries where there's no legislation at all. So I think it's again, it's not my field. I'm not into regulation and regulatory issues so much, but my understanding, I mean, to my pleasant surprise, outside Europe, it's been quite open and flexible. In fact, the UK now is considering regulating gene editing differently. There's a consultation going on right now by the government, the post-Brexit UK, you know, and so it's so yeah, it's interesting. There's I think a general drive to sort of more loosely regulate gene editing compared to transgenics. We should also keep in mind something, you know, there's an important aspect of this discussion, which is traceability. If you want to regulate anything, whether it's a chemical or biological process, you have to be able to tell that actually the product is modified, is synthetic, etc. And one of the issues with gene editing is you could actually introduce mutations through the method that are identical to the to naturally occurring mutations. You can actually recapitulate a mutation that is occurring in one variety in a different one using gene editing. And in that case, it's really hard to trace. So I really, considering that many countries are loosely regulating gene editing, it's very hard for me to know that in 10, 20 years, how some countries would be able to actually prevent sort of gene-edited products, food, and so on, entering their food chain. I really find that I don't know how they would do it. So there is actually a big dilemma in regulating gene editing because of traceability. If you cannot trace it, it's very hard to regulate it.
Okay. One from Peter Werner. Can we do gene editing in the mitochondrial genome? Maybe to introduce male sterility. Okay.
Yeah, I can think of a paper right now, but I don't see why not. Yeah, no, absolutely. It actually is also being done in bacterial research, for example, now. So it's, yeah, no, it should be done. Yeah, it can be done. I don't have a paper in mind, but yeah, I'm sure it can be.
Okay, and another phone from Mr. Siddiqui from Pakistan. Hello. CRISPR edited are not GMO in his opinion, but would these be considered genetic integrity, same for conservation?
I'm not sure what you mean by genetic integrity. Let me simply say that I think one of the challenges right now, and one of the, I think one of the question marks I would say on the use of gene editing is whether there are genetic rearrangements that are occurring in the genome following the CRISPR method, and there hasn't been yet, I think, a fully robust answer to that. Particularly because of the technology currently, the genome sequencing technology is not that good at finding big structural changes compared to the specific changes in the genome. So so that remains to be answered.
Okay. Again, another question from Babar. Sometimes Cas9 sequences, small in parentheses, integrates to the host genome, which are difficult to locate using simple PCR. How do you overcome this problem?
Well, I think there's an answer in the chat. So thank you, Abdullah, for the answer. It's exactly the answer is whole genome sequencing. And it goes back to the comment I just made. So whole genome sequencing currently is very good at finding changes in the sequence, but it's harder to find structural variants. So if there was a big rearrangement in the genome, that's actually a little harder to find right now by genome sequencing. But if there are additional mutations or fragments of Cas9 and so on, that can be easily detected.
Okay. From Thomas Payne in CIMMYT, to what extent is mutation breeding or CRISPR in ornamental horticultural crops or in microbiology, fungi, bacteria, in other words, in crop systems that are not used for direct food consumption?
Yeah, absolutely. I mean, again, one of the incredible thing about CRISPR is that pretty much every organism now can be CRISPRed. And there is actually a gene-edited mushrooms. I think they were commercialized in Pennsylvania, which are they're resistant to the browning, so they last longer. So yes, multiple applications in many fields, not just plant biology, definitely.
Well, it could also be
in
mutation of a pathogen that is attacking a crop, right? If there is a virus or a pathogen,
yeah. I mean,
you could mutate that bacteria so that it doesn't attack, anthracnose or whatever. You could mutate that rather than the crop itself.
Yes, absolutely, Annie. And there is also this concept of gene drives that I didn't get into, which CRISPR can enable, where essentially it's like if some of you who are entomologists would know about male sterility and so on, and so introducing essentially sterile populations that would sort of kill off the population of the pest or pathogen. And so one version of that is so-called gene drives, where you introduce a gene that, this is a bit controversial. There's a lot of discussion about, the potential side effects of that, but this is certainly a possibility that could be considered in the future.
Okay, we're nearing the hour. There's 3 more questions. Are you still up for it?
I'm fine. If
Badara,
up for it,
Badara
good discussion. Thank you.
Yeah, not sure CRISPR will be useful to gene banks if the notion of genetic purity and integrity is the major principle of plant genetic resource conservation. So it's an opinion, but I think he's asking you, what are your thoughts on that?
It's an interesting opinion. I think it's It's an interesting point. I'm all for conserving pure and again local varieties, land races, and all that. I think the question now is first of all, a lot of those are also the product of mutation breeding. So in a way, that's technology that's not very different from CRISPR. I think the question that arises now is whether you want to also conserve all the mutants and variants that come out of CRISPR. I mean, that's also valuable to conserve. Like I showed you that dwarf tomato, right? So to me, that's actually fairly rare a mutant, a very, very specific mutant with only those 3 nucleotides deleted. And that's, that's possibly not super easy to generate again. So I guess that's the type of material you want to also conserve. And we are starting to get organized in terms of having. I had also a prepared slide for you. I was expecting a question along this line, but here's a database that Greg Martin at Cornell University has put together with funding from the National Science Foundation. And I think, as far as I know, this is the first public database of CRISPR mutants. So, we obviously think that those mutants are certainly valuable at the moment for research, perhaps primarily, but possibly also for plant breedings very soon. And what I showed you there with Biogel in Changzhou is also essentially what they have is really a gene bank of sorts. So I think it's complementary. I really don't think we should look at these technologies as competing, but rather complementary to each other.
Yeah. Thank you. A question from Nora Castañeda. How feasible is it for a small, medium-sized seed company to take over this technology in its breeding program?
It's a very interesting question, and there are indeed companies right now. I think the biggest challenge they have is really IP, and it's complicated, as you can imagine. But there are startups, for example, that have alternative CRISPR system or alternative gene editing system, and that certainly is a good basis for a company, for a startup that could be disruptive. But your question is really pertinent. I'll tell you why. Because one of the problems with transgenics, with the classical GMO, let's say, approach, is it's so expensive to deregulate that only few multinationals really have access to this technology. I mean, and now with CRISPR, essentially anyone can do it. Even a lab in a developing country can generate their own CRISPR mutants. So you saw the Biogel also case in China. So I think that's indeed, there is potential for having here disruptive technology for the multinational, the ag multinational companies that really fully dominate the transgenic market.
Good. Our last question is from Tom Payne. Since you're in England, what is the mood for use of CRISPR in Brexit UK?
Well, it's an interesting question, very timely, very opportune, because currently the current government is favorable for making CRISPR sort of more loosely regulated, let's say, certainly not compared to transgenics. And there is a consultation going on right now by the DEFRA, the Department of Food and Rural Affairs, on how CRISPR, how gene editing should be regulated, and it's not just plants, but it's also livestock. So it's a broader discussion, and I guess the push from the government is to more loosely regulate and open up this this technology and sort of maybe placing themselves more closer to the US kind of situation than to the continental Europe or the European situation. So the government mood is along those lines. I can't really tell much about the public mood, but my feeling is that currently, I think globally, I hope that there is more of a pro-science attitude in the world, especially with the COVID situation and with the way science, not just through the vaccines, but also through genome technology, is really helping us deal with this problem. So yeah, I don't know, but certainly there is I mean, it's being set in motion to more loosely regulate gene editing in the UK.
Okay, well, thank you very much, Sophien. We really enjoyed your presentation and your candid answers. Thank you for taking the time to prepare a talk and all of that. We know it was a little bit distant from what we normally do, but we really appreciate that you came on as the second presenter in our Grow webinar series. To all our attendees, thank you very much for logging in today. We will be again having another speaker. 2 scientists from USDA will be presenting in March, 18th of March. So we look forward to having you join again next month. And to Luigi, Linus, and Faye, thank you so much for supporting us. And everybody have a great day. Stay safe. Bye-bye.